Hello, my name is Josh Mahlios, and I'm a Senior Marketing Product Manager for Miltenyi Biotec. And thank you for joining this webinar that focuses on the latest technologies available to reliably isolate top quality starting materials for your drug discovery research. The agenda for today is as follows. First, I will briefly provide some background information about Miltenyi Biotec. Next, we will discuss the importance of understanding the various starting materials used in drug discovery. We will then talk about the various isolation methods available for the generation of PBMCs and primary immune cells, and this will be followed by highlighting the advantages of automated cell isolation protocols. Finally, I will provide the latest resources available to support your drug discovery objectives. The words empowering discovery and advancing therapy really define our company's mission, which is to improve scientific understanding and medical progress by providing solutions that advance biomedical research and cellular therapies. As a company, we are able to achieve this mission by our ongoing commitment bringing newer and smarter technologies to drug discovery scientists who are paving the way for longer and healthier lives. Miltenyi Biotec was founded in 1989 by Stefan Miltenyi, and the basis of the max cell isolation is the column technology developed as a part of his diploma thesis, with this technology being patented in 1990. The potential for max technology to become a revolutionizer in the field of gene therapy became obvious, and the CliniMax CD34 reagent system for selecting hematopoietic stem cells from donor apheresis was developed. The clinical evolution of the company has continued for over 20 years, including setting up a GMP manufacturing site in Federal, Germany. And in 2014, the FDA gave approval for CD34 isolation using the CliniMax together with the CliniMax CD34 reagent. What started out as a diploma thesis has since grown into a global company of more than 3,000 employees, as well as over 20,000 scientific publications citing MEX technology. Annually, more than 2,500 patients receive cell products manufactured with Miltenyi Biotec technologies, thus allowing us to recognize our company mission of advancing biomedical research and cellular therapies. So what is MAX technology? MAX technology is based on three key components: the MAX microbeads, the reagent, MAX columns, and the MAX separator, the magnet. Together, they enable the magnetic separation of cell populations based on surface antigens. This technology is a fast and gentle method for the isolation of viable and functional cells by labeling epitopes with specific antibodies conjugated to superparamagnetic beads. There are only three easy steps necessary to generate viable cells for drug discovery using MAX technology. First, you have the magnetic labeling, where an antibody that's conjugated to a tiny superparamagnetic microbead that binds to a surface receptor or protein on the cell surface. You have the magnetic separation, where the cells are passed through a column that is positioned within a magnet. The cells labeled with the microbeads are retained within the column, while everything else is washed through in a gentle way. The flow-through is called the negative fraction containing the cells without the ligand for the antibody or antibodies that were used. In the elution step, step number 3, the column is removed from the magnet, enabling the magnetically labeled cells, so the positive fraction, to be washed out. This can be done either manually with a plunger to gently push the cells out, or it can be automated using a vacuum to pull the cells out, or increasing the flow speed of the buffer in the system. All three methods are gentle for the cells and separate the cells into the negative and positive fractions with exceptional purity and high viability. A frequently asked question is, why do we use columns in the first place? The magnetic separators in the column-based and column-free systems have a comparable magnetic force, but the real differentiator between the systems is the column. This is filled with small iron spheres, and each iron sphere passes the magnetic force onto the next one over a very small distance, thus amplifying the magnetic force by nearly 10,000-fold. This ensures that the magnetic field is constant and strong throughout the column. This system allows for the minimal labeling of cells with microbeads to retain the target cells in the column without relying on epitope saturation or large beads that can interfere with downstream applications such as cell culture or flow cytometric analysis. The iron spheres have a cell-friendly coating that allows the cells to run freely through the column and can be washed thoroughly to remove cell debris and contaminants. All this contributes to the fact that the MAX column allows for highly effective cell separation with minimal effects on the cell. Originally starting with MAX technology 30 years ago, Miltenyi's product portfolio has grown significantly, and the translational approach from benchtop to bedside is reflected in all of our products. We provide many of the same products in both research grade as well as GMP and clinical quality to ensure smooth transition into the clinic. We provide a comprehensive offering of products for drug discovery workflows. This includes tissue dissociation and homogenization, cell isolation and culture, including the activation and expansion of target populations, as well as the instrumentation and reagents for cell analysis and tissue imaging. Now, as you might have noticed by now, everything at Miltenyi Biotec is centered around cells. And this brings me to a critical question for those attending. Where do you normally get primary cells from, and how do you process them for your downstream assays? To address the first question, we will talk about sourcing blood products as a starting material. So let's look at the common materials used for obtaining primary cells and the steps involved. From your starting materials to sample preparation, followed by magnetic cell isolation and downstream applications. Every step is critical to ensure consistent and reproducible downstream application outcomes. While primary cells can be obtained from various tissues, most human primary cells come from blood products such as leukopaks. There can be a fair amount of flexibility in this area depending on the number of cells needed in your downstream applications. Whether the starting material is from whole blood or bone marrow with limited cell numbers, to buffy coats or leukopaks, which are larger in size and provide substantial amounts of quality starting materials for drug discovery assays. Most commonly, these are PBMCs, and in the next slides, we'll talk about PBMC isolation. So let's explore the main sources for PBMCs. Whole blood is arguably the most common starting material used globally, where you can find between 1 and 2 million PBMCs per mil, with the volume being variable depending on the donor, and frequently provided in different anticoagulants based on the needs of the end user. The amount of hematocrit is rather high, typically around 40 to 50%, which can have an impact on cell separation, preparation, and downstream assays. While the PBMC concentration in whole blood is rather low, buffy coats contain on average about 500 million cells in a volume between 30 and 80 mils, with 50 mils on average. A buffy coat is a waste product generated from whole blood donation, and the hematocrit levels vary between 4 and 10%. The same hematocrit levels can be found in LRSCs, leukocyte reduction system chambers, or also called buffy cones or trema cones, depending on where you are. These cones contain on average a billion PBMCs in a rather small volume of only 10 to 15 mils. Similar to buffy coats, LRSCs are a waste product, but in this case are a waste product of platelet pheresis. The highest PBMC concentrations can be found in leukopaks, and they contain up to 10 billion cells in a full pack, and the volume varies typically between 80 and 200 mils, but sometimes can be up to 300 mils depending on who your provider is. The hematocrit levels are typically very low and are less than 3%. So as you can see, there are clearly a lot of differences between these starting materials, and it's important to know exactly which material you are working with in order to use the right cell isolation product and protocol. The importance of top quality starting materials cannot be overstated, as experimental success and reproducibility are contingent on this aspect of drug discovery. Whether you're using fresh, frozen, or mobilized leukopaks, whole blood, bone marrow, or directly sourced PBMCs, Miltenyi Biotec has become a source for the most reliable and consistent blood products on the market. With multiple collection centers throughout the United States, we provide an extensive donor pool that allows you to choose from products based on donor characteristics and recallability. For more information about obtaining blood products from Miltenyi Biotec in the US and Canada, please reach out to your technical sales consultant for ordering details. Once you have obtained the necessary starting materials that meet the quality requirements of your downstream assays, it's necessary to process these cells to obtain the cells of interest. Let's take a look at the various processes that exist to do so. Typically, the sample preparation step when using blood products such as whole blood or bone marrow, buffy coats, leukoplaques, or LRSCs is traditionally a density gradient centrifugation step to get PBMCs. Now we'll talk about this step in a bit more detail in just a minute, but first I want to walk you through the rest of this generic process. Now, once you've obtained your PBMCs, you would proceed with the steps that we had previously discussed for magnetic cell separation. And depending on the project that you have, different options to select from. Would you rather go for an easy and fast positive isolation? Or do you need your epitopes free for a second magnetic isolation so that you prefer an untouched isolation where you label everything else except for the population of interest? Do you have multiple samples and would prefer to separate them in parallel compared to one after the other? Would you rather freeze aliquots of your PBMCs or isolate the cells directly? All of these factors are important in deciding which strategy is right for you at this step. Once you have your population of interest, you can then move on to downstream applications, whether it's an additional magnetic cell separation of subpopulations such as Tregs, or the flow cytometric analysis necessary for immunophenotyping. We can recommend the kits that will make these steps easier. Perhaps you're culturing the cells in vitro assays or are running genomic or proteomic applications. Whether you are interested in maximum recovery for cell culture assays or high purity for reliable genomics or proteomic studies, we can help you with the strategy to get there. In order to accomplish these objectives, you need top quality starting materials. And for the rest of this webinar, we will be focusing on the latest advancements in the generation of PBMCs and primary cells from blood products. Now we're going to take a look at a protocol for isolating PBMCs from various blood products. So I'm sure I don't have to tell you what density gradient centrifugation looks like. And without going into too much detail, there are a lot of steps. You have to layer, centrifuge, count cells. It takes a lot of time and is prone to human error and user variation. Fortunately, we have developed new products that will allow you to bypass the lengthy density gradient protocol and get the cells of interest quickly and reliably. Miltenyi Biotec's density gradient-free solutions allow you to isolate cells directly from blood products without density gradients, without cell counting, and only in a few simple steps. You can completely skip the density gradient centrifugation. The protocols are very straightforward, and it's easy, and you have the full flexibility in terms of isolation strategies. At the same time, these are the fastest protocols available for the isolation of PBMCs and immune cells directly from the different blood products that we've discussed earlier. There are very little hands-on steps, and user variation can be reduced significantly due to the automated options that you can choose from. When discussing density gradient-free solutions from Miltenyi Biotec, I'd like to first start out with our latest offering that allows you to isolate truly untouched PBMCs from blood products. Depending on your starting material, we have specific isolation kits available for the untouched isolation of PBMCs from peripheral blood, buffy coats, LRSCs, and leukoplaques. The workflow between these starting materials is almost identical. So let's just have a look at the protocol in general. First, we sediment the erythrocytes using a dedicated sedimentation kit. Including the pipetting, this only takes about 5 minutes. Afterwards, we take the supernatant and centrifuge for 5 minutes. After resuspending the cell pellet, we can directly continue with labeling of the cells with our max microbeads by adding the reagents, and we incubate for 10 minutes. Next, the magnetic separation will be done in our cell separation instruments, which takes between 5 and 20 minutes depending on your starting materials and the volumes that you're working with. Our protocols ensure that you will only spend between 25 and 40 minutes to obtain untouched pure PBMCs from your blood sample. When comparing the processes for generating PBMCs, we can now take this one step further and compare the total time it takes from your samples to PBMCs. We've seen how long that density gradient centrifugation takes, no matter what your starting material is. Of course, it also depends on the volumes, the dilution factor you work with, and therefore the number of tubes that you need to handle. But just to have an example, it definitely takes more than 1 hour and possibly more than 2 hours to process PBMCs using density gradient centrifugation. This is what you can see here in purple. Now, when we compare this to our new PBMC isolation kit, we immediately see that the protocol is much faster, independent of the starting material. When working with whole blood, buffy coats, buffy cones, only 10 minutes of hands-on time is required for the preparation and sedimentation step. This means that sedimenting RBCs and taking up the supernatant and spinning for 5 minutes is all it takes. Afterwards, the process, including the labeling and separation, is fully automated. For leukopacs, the initial sedimentation of RBCs and the centrifugation of the supernatant can be skipped, and we can immediately start with the labeling, followed by magnetic cell separation. Without going into the details, this slide is showing how the composition of PBMCs is not altered from the starting material when compared to traditional density gradient centrifugation processing. This should give confidence to the end user that the technology doesn't selectively deplete any potential subpopulations of interest for further downstream assays. Now that we have covered our latest untouched isolation methods for PBMC generation, we will now turn our attention to our straight from portfolio that enables the fast and efficient isolation of multiple cell types from the same blood sample. Similar to our untouched PBMC isolation products, the protocol for straight from is very similar, independent of the specific starting materials that you work with. No matter if we are talking about whole blood, buffy coats, LRSCs, or leukopacs, first you fill the blood sample into a collection tube. Next, straight from microbeads are added, and then you incubate for 15 minutes in the fridge. Then the magnetic separation is done on the Multimax separator, or in the case of whole blood, you can also use the AutoMax Pro separator. In both cases, it takes just 30 minutes from the blood sample to you holding your target cells in the hand. Straight from is based on a positive isolation approach from the starting material, and this provides a very attractive advantage. This advantage would be how positive selection enables consecutive separations from the same sample by using the negative fraction for another round of separation. Here we have an example. First, you isolate CD8 positive T cells and then take the negative fraction for isolation of CD4 positive cells. You could take this even another step further and isolate another round of subpopulations from that negative fraction. Therefore, if you're interested in several target cell populations from the same blood sample, this may be the ideal product for your experiment. A frequent question that we encounter when discussing our positive selection reagents has to do with the performance and downstream functionality of the cells that are isolated. In this case, we've checked for T cell enrichment, T cell activation, and transduction efficiency as compared to density gradient centrifugation. And as expected, we have comparable results. But of course, the T cells and the separation were done much faster using the straight from approach. This means that our isolation will allow you to choose the cells of interest with no negative side effects on your downstream results, but allowing you to be more efficient at the same time. In this next section, I will talk about automating your cell isolation workflow. Now, why do we actually need automation? What is the advantage? As we've already said a little earlier, automation reduces hands-on time and frees you up to work on other tasks in the meantime. That can't be automated. It minimizes the variation between different users in the lab, or even a good or bad day from the same individual. And of course, it minimizes costly human errors. With standard protocols and minimum user variation, you can be sure that the effects that you're seeing are due to your assay rather than artifacts. In short, Automation reduces redundant experiments and allows you to focus your time where it matters. When introducing our family of automated cell separators, we'll start with the AutoMax Pro, the easy-to-use benchtop instrument for every laboratory. With fully automated sequential separation of 1 to 6 samples, the AutoMax provides easy access to the automation without high investment costs or lots of training. You can mix many different starting materials and samples within one run, and just hit the button and the instrument will label and separate the cells for you. The columns are reusable, so maintenance is easy and column costs can be reduced. The instrument is the perfect match for the isolation of PBMCs from peripheral blood and buffy coats, but of course also target cells that can be isolated from PBMCs within the AutoMax Pro. If you are interested in isolating multiple samples in parallel, the Multimax family is a perfect fit for this. 24 magnets allow you to process between 1 and 24 samples in a single run, or split your large samples between multiple columns. This is achieved by using the LS, LD, or whole blood columns, or the multi-24 column block for easy handling of 24 samples, or perhaps a very large sample, like an entire leukopak. The Multimax Cell 24 is a semi-automated instrument that is a good fit for many labs that value easy handling but don't mind the manual pipetting. However, if you want full automation and maximum throughput, you will love the Multimax X, which integrates the Multimax into a liquid handler. The Multimax X is the instrument for the high-demanding, high-throughput cell processing lab. Samples are labeled and isolated automatically, both for PBMC generation or subpopulations from all blood products. All programs are tailored to match your needs, so you can be confident that you will be satisfied with the protocol and the results delivered from this instrument. So, we are coming to the end of the presentation, and there are a few things that I want you to keep in mind when thinking about density gradient-free solutions for cell isolation with MAX technology. It should be no secret that the success of any experiment depends on the quality of the starting material. So remember that the right cell preparation and isolation steps are crucial in the beginning of any workflow to ensure reliable assay results. Automating these processes with the help of our cell separation instruments, combined with our density gradient-free protocols, allow you to be more efficient while ensuring high reproducibility and standardization. Our new methods enable you to isolate target cells or PBMCs directly from blood products with high speed, great performance, and easy protocols. I'd like to thank everyone for attending this webinar, and for the latest updates and resources available for drug discovery workflows, please come by the booth or stop by our website. Thank you and take care.